Hi everyone, good afternoon, and I'd like to thank Hagit and Roy, which I understand are not here, for the invitation, and all of you for coming. So they asked me to give quite a wide perspective lecture about the field. So what I will try to do is to present our field of interest, which is most of the lab is working under the topic of the neurobiology of stress. So I will give quite a bit of introduction, mainly explaining the rationale, why it's important to study these type of questions, and then focus on the methodologies we are using in order to tackle a specific question in this field. As you will see, it's very much oriented into mouse genetics, and I know the background here is very diverse, so I will do it relatively slowly. I tried not to put too many details on each project. I prefer to show you the different approaches and then the take-home message from each project. So, of course, feel free to ask questions in between and everything. So, as I mentioned, the neurobiology of stress is the main focus of the lab, and we refer to stress as the result of any challenge to homeostasis, and it will be clear in a few minutes. As you know, following any stressful stimuli, whether it's going to be psychological or physiological stressors, multiple brain regions will be activated. Those brain regions associated with emotion, with cognition, with locomotion, with metabolic function, neuroendocrine function. Many brain areas are going to be activated when a lion is just going to enter the room. However, the way this activation is eventually integrated into a final common response in the brain is not fully understood. Nevertheless, we know when this response, when it's not properly regulated, and I know regulated is a big word, but what I mean by regulation, and you will see it more and more, it's when it's not properly turned on or mainly turned off on the right time. When this is not a lion, this is alone in the custom of a lion, the system has to be switched off, and when it's not properly regulated in the gene level or the cellular level, this eventually can cause different stress-related diseases, and when we talk about psychiatric disorders, we talk about anxiety, depression, and PTSD, post-traumatic stress disorders, and eating disorders, but not only psychiatric disorders, also heart-related diseases and metabolic-related diseases, and of course, more and more. So what we are trying to do is trying to dissect, and this is the title I gave, is to dissect the contribution of very specific genes, proteins, brain circuits, in both the pathophysiology, but also in the normal physiology of the organism as part of the stress response. So what we mean when we say stress response, you understand what the stress is and what are stressors. Yeah, a lion could be a stressor, but of course, also when I have an important exam or a contract, a grant I need to send tomorrow and have a deadline, it's definitely a stressor. But the stress response will be this series of coordinated responses which have two main phases. One will be survival, okay? It's a challenge, it's a survival response, it's a very conservatory evolution. If I have a lion here, the first thing I want to do is to survive this challenge. And the second step of the stress response is to restore homeostasis, okay? After, as you will see in a minute, activation of the stress response will move most of the system in the body out of homeostasis, that I will be able to cope with this challenge. And then the second step, when the challenge, when the threat is not here anymore, I need to switch off the system and go back to homeostasis. And these two parts are very important. So biologically, we talk about activation of two main systems. One is the activation of the sympathetic nerve system, or the catecholamine release, or adrenaline, and activation of the pathologic pituitary adrenal axis, which I'm going to explain more in detail. Of course, these two systems have a major crosstalk between them in the brain. Okay, meaning ligands from this system will activate receptors or cells which express, for example, 5-HT, and I will show you examples for this. So activation of these two systems, which are simultaneous, simultaneously happening will lead to a long list of central and peripheral effects. But all these changes, and just listed some of them here, will work to the same direction to allow you to cope better with this challenge. Okay, of course, you're going to have changes in your behavior. You're going to have changes in the level of your anxiety, your fear level, your memory is going to change, your attention, your focus attention is going to change. Okay, if by average it will take us 20 seconds, all of us to notice the lion standing next to me here, after all of us notice the lion and perceive it as stressful. Of course, if a tiger will enter now, it will take us, I don't know, a second, even less, to notice it. So the attention has been changed. Of course, you're going to have changes in the cardiovascular output and respiration, increased heart rate and blood pressure, of course. Cerebral perfusion going to change. The CSF going to, both the rate of the CSF and the context is going to change. You're going to have increase in your blood glucose level from 100 to 160 or 70 in a few seconds, just in order to be able to cope. Or if you need to run, that the muscle will have enough energy in order to run from this line. Of course, you have modulation of the immune function, decrease in feeding and appetite, or decrease in reproductive function. Of course, the last thing I want to think now when I have the lion here is about my dinner, about sex, or anything else. So you have inhibition of some of the system, activation of the other system. And the reason I put so many different systems is to make a point that when the system is not properly regulated, each of these systems could be affected. Okay, this is the reason you can just Google stress and almost every disease, and you will find a link. You will find something. The problem that in most of the cases you will not find mechanistic insight. That you will have epidemiological evidence. You know, stress and cancer is related to stress, and disease is stress. And so some of the system you have better understanding of the mechanism, and what we are trying to do, of course, as basic science, is to get more and more understanding of the basic mechanism, which is mediating these responses. We focused mainly on one of these systems, which is, I mentioned it before, which is the HPA, this system. And the most known peptide activated, which is responsible for activating this axis, is called CRF or CRH, which stands for corticotropin releasing factor. So following any stressful stimulation, this peptide, which is relatively short, 41 amino acid neuropeptide, is going to release from its hypothalamic neurons. It will reach the pituitary gland, will bind this receptor, which in response will release hormone into the blood circulation. This hormone called ACTH, or adrenocorticotropic hormone. This hormone will reach your adrenal gland on top of your kidneys and will bind this receptor in the adrenal cortex. In response, will release cortisol or corticosteroid in rodents into the blood circulation. So, cortisol, corticosteroid have multiple effects in the periphery. Almost every organ, every cell have glucocorticoid receptors, which, by the way, this is the Wolf Prize for this year. And I think Ron Evans is going to be here in a few weeks receiving his prize for discovery of these receptors. But in addition, they're responsible for this classical negative feedback. They're going to enter the brain, and they're going to reduce the expression and secretion of this peptide and this hormone into the blood. In addition of having a multiple function inside the brain. But CRF is not playing only as a neurohormone, activating this peripheral cascade. It's also working as a neurotransmitter, as a neuromodulator inside the brain. And it's highly expressed in the amygdala, in the cortex, in the cerebellum, in the hippocampus, that we'll see in a minute, where it mediates. At the same time, you release by these hypothalamic cells and activate the periphery, but also release as a neurotransmitter in the brain is affecting stress-related behaviors, such as arousal, anxiety, fear, memory, and so on. So this peptide had already been isolated in 1981, and it was probably the most studied peptide ever if you search the PubMed. But following the completion of the Genome Project around 2001, 2002, realized that CRF is actually part of a much bigger family of peptides in the brain. And we currently have 4 different peptides. They are called the urocortins. We have 3 of them. We call them urocortin 1, 2, and 3. All of them are 38 to 41 amino acid neuropeptides. They encode by different genes. They will see that located in different brain area. Their effect is actually mediated by 2 receptors. These are G protein coupled receptors, 7 transmembrane domain receptors. Both of them are coupled to GS, if those of you doing some signaling. This is the receptor which is responsible for this HPA axis I mentioned, the hypothalamic pituitary adrenal axis I mentioned. But it's also widely expressed in the brain. The pharmacology of these is actually relatively very interesting. CRF have high affinity to this receptor, very low affinity to this. This is just in the test tube, okay? We'll go into the brain in a minute. Urocortin-1 have high affinity for both receptors, while urocortin 2 and 3 are specific ligands for this type of receptor. So in addition to the 4 ligands, 4 peptides, and the 2 receptors, we also have 2 secreted binding protein. One of them is what is called the CRF binding protein. I will get in the end of my talk for a lot of time. I will mention it, but this, during pregnancy, the placenta is generating huge amount of CRF. For many years, physicians and scientists couldn't understand how women cope with such high level of stress hormone in the blood, which eventually led to the isolation of this protein, which is a binding protein, which actually plays a decoy. It's actually holding the peptide and preventing from activating the receptor. And this is another binding protein, which is alternative splice form of this receptor, which I didn't get into. It's a multiple isoform and so on, which is less important, but is also serving as a binding protein. Okay, so the system, of course, is much more complicated than initially thought, and this is just without even showing you the neuroanatomy or the distribution of these different members of the family. What I do showing you here is just the sagittal section of the rat or mice brain, and just showing you the expression of the distribution of the receptors. Okay, so in yellow you can see the type 1 CRF receptor distribution. So in addition of being expressed by the anteropituitary, which is regulated HPA axis or activate the HPA axis, as you can see, it's widely expressed in the brain. In the cortex, specifically layer 4 of the cortex, in the Purkinje cells, in the cerebellum, in the amygdala, and septum. In most of these brain regions, we don't really know the function of these genes. Okay, you need to understand that all this stress field historically started by neuroendocrine people. So those people concentrated mainly in the hypothalamic region. Then they went into more emotional-related area for fear and anxiety. So the amygdala and the bed nucleus and this type of area are more studied. But if you will ask, what is the role of this receptor and all this system in layer 4 of the cortex, no one did it. Okay, so our current study is dealing more with this brain area. We just generated mice, which is knockout, which is specific to layer 4 in the cortex, or specifically in the granulosa, in the cerebellum and the hippocampus and other brain area, in order to try and understand what is the specific role of this receptor in specific brain region, which of course, again, stress is not affecting only our stress hormone, our anxiety level, it's also affecting our attention and other cognitive function and so on. So, one of the major tools in the last 10, 15 years was to generate, tools to study the role of novel genes. Of course, was to develop knockout mice model. Okay, just to delete this gene and ask, okay, what's wrong with my mice? And the last few years, and I don't have time to go to the phenotype of each one of these knockout, but currently we have a knockout for each member of this family done by many groups. I did knockouts for these when I was a postdoc, but in my lab at the Weizmann, we started doing what we call multiple knockout approaches. Okay, what I didn't mention is that the fact that this receptor could be activated by these 3 ligands, this is affinity in the test tube. But if you look on the distribution of the brain, we know pretty well which peptide is activating or is innervating which receptor and where. Meaning, if you look on the type 2 CRF receptor, for example, in the dorsal-raphe, which we know it's 5-HT, it's serotonin neurons, we know it could be activated only by urocortin 1 and 2. We know it's not innervated by urocortin 3. So we map the brain very well using antibodies and in cytohybridization, which gene, which peptide is innervating which receptor and where. So I'm going to show you only, I'm going to show you only two mice, one mice model before going into more specific genetic approaches. And what we did recently is generating what we call a triple knockout mice, in which we deleted all the 3 urocortin genes. Okay? So these mice is lacking urocortin 1, 2, and 3, meaning avoiding any activation of the type 2 CRF system. Okay? So what's wrong with these mice? So we went and did a wide battery, and this is something some of you are familiar with the lab. But what we established a very wide battery of behavioral and physiological tests. Okay, some of you saw our new facility, which is like a phenotyping center, which we have a very wide, large number of different tests, which you can measure anxiety, like depression, like behavior, different memory tasks, but also a lot of physiological related tasks, such as metabolic profile, and so on. Those of you which are less familiar with this type of anxiety test, I will not go into the details. There are different tests. I'm sure you heard about open field or elevated plasmase and light-dark transfer test. Some people do it here. But the concept is very simple. Okay, this is what we measure is the conflict the mice have between exploring new environment, which is something you like to do, versus staying in a safe, dark, familiar compartment. Okay, so very aversive, very strong light normally. So all these tests have same concept, but different setup, different intensity of stressful environment. So when we take, and this is open field test, it's not really important what the test is, but when we compare the triple knockout mice with their wild-type littermates, without any stress, we just took them, we put them in the test. The test is a bit stressful because it's a new environment. Still, we didn't see any differences in the anxiety level of these mice. Then when we took another group of mice, and this time we stressed them. Okay, we took them, before the test, we did what we call a restraint stress. We put them in a restrainer for 30 minutes, very stressful for the first time, and then immediately after, we put them in the test. We still didn't see any differences, regardless, meaning both the wild type and the knockouts were more anxious than the previous one, but both of them were anxious the same way. But then what we did was something else. We took another group of mice, we stressed them for half an hour, but instead of testing them, we put them back into the home cage. And then we came back 24 hours later. Okay? And then we saw major differences. This is regardless of the type of test we do. Okay? And the differences are very clear. And what are the differences is that the wild-type mice, after these 24 hours, they came back and they coped properly, meaning they went back and have normal anxiety levels. But the triple knockout mice just didn't recover, and they continued to show high anxiety. And this is one day after, but if you come even 1 week and 2 weeks after a single acute stress, which is not very traumatic stress, it's stressful, but it's not traumatic stress, these mice are still extremely anxious, suggesting that the role of this urocortin, this type 2 CRF system, the right part of this diagram I showed you, is very important for coping, for recovery from stress. And as I mentioned, one of the strongest theories today in the field of stress that people will develop stress-related psychopathologies when they have problem in switching off the initial response to the stressful event.
What do people know about these 3 types of urocortins before your study?
So again, a lot of it is some of it I did already during my postdoc, but it was mainly based on the individual knockout. And I will go very quickly to the problem with the previous studies, and this is the problem with developmental knockout when I get into it, because we have individual knockout for each one, and each one have a different phenotype, which was very confusing. And of course, we also have a lot of pharmacological study. But the concept is, the theory is this system, this part of the system is actually activating the stress response, and this part is actually switching off. And this was actually raised already a few years ago. But the problem is that pharmacological studies not always agreed with this concept. Okay, so this is one of the things we did in order to try and see what is the direction. It indeed looks like coping, But as you will see, when you start doing specific manipulation, it's not always the case. It really depends where in the brain, in what context, what peptide, and what concentration. So it's very complicated. But this is definitely put a stamp on the concept now that this is a coping-related system. So how it's mediated? What we did here, I know it's a complicated slide, but I'll walk you through it. What we did, we took the wild-type mice or the triple knockout mice, under stress or 24 hours following the stress. Okay, and then we punched out the amygdala. Okay, of course, amygdala associated with anxiety, fear, memory, and so on. And we designed, this is a real-time PCR array, which we selected those list of 30 genes which are known to be associated with amygdalar functions. Okay? What you can see here, this is just a logarithmic scale, and each dot is a different gene. Everything above this line is going to be significant. So if you compare the expression of this 30 gene in the amygdala of unstressed wild type versus unstressed triple knockout, we don't see much of a difference, which is very similar to what we see in the behavior. We don't see any differences in the behavior. If you take the wild type unstressed and the wild type 24 hours following stress, you see all these changes in gene expression, even 20-fold difference in gene expression. Going to be 5-HT receptor and GABA subunit and dopamine receptor and CRF receptor and so on. Most of them, if you go into the literature, you do PubMed, you will find out that these increase or decrease in the expression of these genes are necessary for coping. It has already been done before. But if you do the same experiment on the triple knockout, again, comparing unstressed triple knockout in 24 hours following stress and triple knockout, all this effect is blunted. Okay? Which may suggest that the urocortin actually upstream to all these changes. You with me? Okay? They're actually may be responsible for, actually we now may have a target that we can approach pharmacologically and try to improve coping in a way. Again, I'm showing you again a very small part of this study, and you'll understand in a minute why it's still problematic. Something else we did with these mice and other mice is trying to understand, as I told you, these two systems have a strong crosstalk between them. And the receptor for this urocortin is highly expressed by the dorsal raphe. Okay? So what we did in this study, just a diagram, but what we did, we measured the 5-HT and its metabolite, 5-HT1AA, levels in the dorsal raphe, in the production side, but also in the target site, okay? In multiple target sites, which what we call belong to the anxiety circuit in the brain, and it was very nice to see that it's correlated with the behavior, okay? The changes we see in the level of the 5-HT nicely correlated with the behavior of these mice, okay? So what is the problem with all this knockout? This is the reason I showed you only one, and I'm sure most of you are familiar with, yes? Yeah.
Did you check the mice for other non-stress-related memory?
Oh, yeah, of course, yeah. This is of course. So each one of these is a very big study, so I didn't show you. We did, of course, different cognitive tests, memory tests. We did depression type. We do, of course, every time. We always do locomotion-related tests because all these tests are based on locomotion. So if you have a problem with locomotion, of course, the mice will show. So we always do locomotion in the test, but mainly also not in the test itself, in the home cage, to confirm that they indeed don't have any. So it's very anxiety-oriented in this case. So what's the problem with these type of models? They have two main problems. One is compensation. As you know, these genes were deleted very early through development, already in the fertilized oocyte. So we delete gene. The brain is, very plastic during development. So you can always, and we did it, we saw it in the other models, individual models. You remove one of the genes, other genes goes up and compensate, other gene goes down. So eventually when you phenotype a mice, you don't know whether you phenotype the consequence of the deletion or the consequence of the compensation. This is one major issue. The other issue is the resolution of these methodologies. Yeah. We all know that the same protein, the same gene expressed by totally different brain region. The same gene expressed in the dorsal raphe, the ventromedial hypothalamus, or the lateral septum, may have a totally different function, and we know that. Not even different. I will show you example, even an opposite function, and I will try to explain why. So by doing conditional knockout, by removing this gene in adulthood, but since we don't have a genetic way to do it in very specific manner, only the dorsal-raphe, only the lateral septum, we have way to do it in the forebrain, in the hindbrain, maybe in multiple brain area. I still don't have the special, the temporal resolution to do it in a very specific brain area. Okay, so when I'm phenotyping the mice, we're getting such an average phenotype of the deletion of this gene by multiple brain area, which could be totally different. Could be increasing anxiety here, decreasing anxiety there. When I'm phenotyping the mice, I'm getting the average. Okay, so we need to develop tools in one way to do it in adulthood and not through development, to try to minimize the compensation. And secondly, to try and do it specifically as possible, meaning to go in and manipulate only this population of neuron, only this and only this. Okay, this is the reason most of the studies in my lab in the last few years were concentrated on using viral tools in order to target very, very specific brain region. And if I can I, this is the light? Yeah. I think it's going to be. Okay. So the advantage of using viruses and you will see lentiviruses or AV, adeno-associated viruses, to do our studies, is the fact that viruses, of course, you will see in a minute, are extremely efficient in infecting neurons. Secondly, it's incorporating how viruses, at least lentiviruses, are proliferating. They're incorporating their DNA into the host cell and using the cellular machinery of the host cell to make their own protein, which eventually assemble into a virus, and this way they proliferate. So these viruses do not know how to proliferate. They don't have the gene. The gene which is responsible for the proliferation has been removed. Okay? But we put in the viruses have only the envelope and the packaging of the virus, and we put inside the virus the gene that we want them to deliver for us. Okay? And then we can inject and we can deliver these viruses very specifically. All the viruses, just one example of viral, which is overexpress CRF. Those of you more molecular people know that we can actually play with the promoter, meaning the region which is responsible or give a specificity of the expression of this. I can use here only neuronal specific promoter or glia specific. I can use only excitatory type of neurons. So I can target where I want this virus not to infect, but where I want him to be active, for example. And of course, all the viruses will express a reporter, okay, such as GFP, green fluorescent protein, red fluorescent protein, which eventually I can track or follow the infected area. This is just in vitro confirmation that every infected cell you can see in green is also immunoreactive for CRF and Western blot. It's less important. So after we do all this in vitro confirmation, this is just to show you the efficiency. Those of you working with culture, this is a cortical culture we did from embryos from E18, E19. Those of you who try to transfect these type of cultures with any transfection reagent, which is extremely expensive, a good day will give you 3 to 5% efficiency. Okay, we're just trying to put your DNA inside. If you use viruses, as you can see here, 5 microliter of virus with high titer will give you 100% efficiency, meaning every neuron in this culture is infected. So the efficiency of the virus is to take the gene, the transgene you deliver inside is extremely high. And again, it's stable because it goes into the genome of the cell. So if I'm going to go and inject these viruses into the brain, it's just going to be there for the rest of the life of the mice. Okay? So we use a computer-guided stereotactic system, which, again, you're familiar with, very similar to any stereotactic, but you have the pathophysiology atlas in the computer. This is very old photo. And then we can deliver using a micropump, we can deliver very, very small volume, half a microliter of concentrated virus in 20 minutes, half an hour, into a very specific, as you can see, specifically into the central amygdala. This is a mice, so it takes a few hours. It's integrated and start expressing. After 2 or 3 days, it will reach its maximum. It will just stay there. Okay, so this way we can gain very specific way to manipulate, as you will see, overexpress or knock down or knock out your gene of interest in a very specific brain region. And then, depending on the brain region you study. So if you did the central amygdala, since we know the central amygdala is associated with anxiety, with fear, with memory, this is the type of studies we do. Okay? Of course, always in the relation of with stress, without stress, acute stress, chronic stress. This is what I mean by dissecting. Okay, dissecting the role of this gene in a very specific brain area. So I will show you a few examples of this without showing you the whole data, just the concept, okay, which is more important for me in this type of mixed audience. So CRF is highly expressed in depressed patients, in the brain, in suicide victim, in the CSF of PTSD patient, depressed patient. The level of CRF is extremely high. Okay, so what we try to do, we try to mimic, and we know it's affecting anxiety and depression, which again have comorbidity between them, but still it's we want to try and understand the contribution of CRF to this type of phenotype. CRF endogenously expressed by the central amygdala and by the beta nucleus of the stereoterminalis. There's a lot of literature, those of you familiar with the Davis studies about the role of BNST versus amygdala. BNST, of course, is what can belong what we call to the extended amygdala. There's a lot of literature about the concept which one is fear, which one is actually more anxiety and not fear. But what we did here, we just used these viruses I showed you before, and we overexpressed the CRF in the central amygdala, or the nucleus, for 4 months, mimicking very long chronic stress in these mice. We didn't stress them again. We just overexpressed it in a very specific brain region. And then we did a wide battery of tests for anxiety and depression. Okay? And what we found is that different parts of the extended amygdala have different role or different contribution to the phenotype. These mice showed much stronger increase in anxiety-like phenotype, while these mice showed much more increase in depressive type of phenotype. And we have different tests to distinguish between them. Okay, and there are other differences, but this is just one of the important take-home message, meaning the same gene in the same extended area, different population will have a totally different outcome. So the previous virus I showed you, in a minute we inject the virus, it's going to be infecting the neurons, it's going to start expressing. So if I want to express my gene in a very small window, not chronically, I need a different system because I cannot stop these viruses. The minute it's infected, they're going to start incorporated into the genome, we're going to start expressing. It's going to be forever. So since we wanted to get a much better temporal expression of these viruses, we used a different approach, and this is combination of 2 viruses. Those of you using molecular studies are more familiar with this. It's called the Teton System. It's what we call a classical inducible system, which use a transcription factor which we don't have in mammalian. But we're not going into the detail, but what we do here, we use combination of 2 viruses. We inject the 2 viruses into for example, into the central amygdala. But these viruses will not start expressing your gene of interest until you will give the inducer. And the inducer in this case is antibiotics. In the drinking water, we just give the mice, we replace the water, we put in the water antibiotics, doxycycline. And the doxycycline is the one which enables these 2 components to interact and to start the expression. Okay? So this is the temporal resolution of hours to days. Okay? I will show you optogenetics, of course, which have totally different type of temporal resolution. But this still could give us hours to day resolution. I can activate the system or switch off the system in 4 hours. Okay? So which give us, again, it's hard to see here with the light. So every cell will be green, but only when I give doxycycline I will see red cell. The problem with this system is what we call leakiness. Those of you who work with this system know that although we see major activation after doxycycline, we still have some cell which is overexpressed without doxycycline. And actually, this new system came out now which we test that people claim it's totally not leaky. but it's interesting. So when we overexpress to very short time in the central amygdala, we get a robust increase in anxiety, which is very similar to what people showed with pharmacological manipulation to this region. It was more important for me to show you the type of technology. So similar to overexpress the gene, you can also knock down a gene. You can reduce the level of a gene. In this case, again, we use viruses, but the viruses we use here is viruses which express what we call short erpine RNA, okay, which is based on the concept of the endogenous ability of this property of the cell of microRNAs to inhibit translation of transcript. Never mind, but we have since the cellular machinery is less simple, what we do, we use 4 different viruses, and we compare them which one is better targeting different part of the gene. But bottom line, if you go in vivo, and this is in situ hybridization of CRF in the PVN, in the paraventricular nucleus, in the hypothalamus, and you can see if we inject it unilaterally here, and this is standing for DAB for the GFP, sorry. So you can see we can dramatically reduce the level of this peptide, and I want to show you one example how we use this type of viruses, and also mention a very important concept in the field of stress. I already mentioned that these genes, which are responsible for activating or switching off the stress response, have to be highly regulated. Okay, this is one very important concept. The other concept is this type of genes could be easily affected by stress during early life. For example, one of the biggest theory of why some individual will be more resilient, or why other individual will be more susceptible to develop, I don't know, PTSD. Yeah, you send a group of soldiers into Gaza or Lebanon, and they will be exposed to the same trauma. Why some will develop symptom of a disease, some will not. So one of the theories is exposure to early life, and then we go into the field of epigenetics, okay, effect or changes in the epigenome of this. And this is what we did in this study. I will show very, very little, small part of this to showing you the concept. What we did here, we used a model of which called social defeat stress. What we do here, we take mice. It's not here. We take mice, and you put them. It's a C57 black mice, and you put them with the bully mice. Okay, it's the ICR. It's a breeder. It don't like other male. It's very territorial. It doesn't like other male in his cage. So you put them together for 5 minutes, and immediately is going and attacking the black mice, which very quickly will do like a surrender posture. Normally, will not really attack them. We would just, and then we keep them together in the same cage with the barrier, okay, with the transparent barrier. They can see each other, they can smell each other, meaning this threat is always there for 24 hours. And then we do it for 10 consecutive days with different bullies, okay? It's considered very strong chronic social type of stress. And then you can even come a month after and do very, very simple tests. And the test is a social type of social interaction test. You take your mice and you put another mice here you never met before, okay? And mice are very social creatures, okay? They like to do social interaction. They always engage in interaction. So if you take unstressed mice, this is just the video tracking of this type of experiment. You can see that unstressed mice will spend most of his time trying to do interaction with his mice. Okay, they have this opening here. They cannot move, but they can see and smell each other. If you take mice to the way in the social defeat, you will see that most of the mice will be susceptible. Meaning they will come and check, but most of the time they will be far as possible from these mice. Interestingly, 10 or 15% of the mice, stress mice, will be what we call resilient. They don't care how much they've been beaten up before. They're going to come again and initiate the interaction. And this is a very, very nice model to study the molecular and cellular mechanism of resiliency or susceptibility. Why some individuals are more resilient and why are more susceptible. And what we found here, and I'm taking you back to the CRF. What we looked here, we looked on the methylation. Some of you are familiar with the methylation, okay? And each box box here is the CPG island. It's a site where a methylation can actually take place. But the black one are the site which is actually methylated. This is the prediction. Okay, so you see the intronic sequence is not methylated at all, almost not methylated, while this region, the promoter sequence, is highly methylated. One of this region is what we call cyclic AMP responsive element site, which is crucial for CRF expression. I don't want to get too much into it. It's a lot of molecular stuff, but we did mutation here. And bottom line is that methylation is extremely important for the transcription of CRF. And I'm not talking about development, I'm talking about adult mice. Meaning, if I'm going to give cyclic AMP to this promoter, the level of the CRF is going to go up by 15-fold, 1.5. If I'm demethylating, if I'm removing the methylation and giving cyclic AMP, I'm going to get 70-fold induction of this promoter. So it's very, very important to the regulation of its level. But what we found, if you take the mice and you isolate this PVN area where the CRF is expressed, and you look on the level of the CRF, you can see the unstressed mice have similar level to the resilient mice, while the susceptible mice have very high level. If you take the same sample and do methylation, you can see that the level of methylation in the resilient is very similar to the unstressed and vice versa. Meaning demethylation of this promoter is the reason why the level went so much high. Okay? I hope it's a very small part of it, but so how are we going to prove that indeed this brain area that we did all this is the one which is important for this behavior at all? And then we can go back to the viruses and use the viruses to knock down, to mimic this effect. Okay? As you can see, again, not go too much into the detail. Without social, we injected the mice with knockdown viruses, with the control viruses. Without social defeat, we don't see any differences. But following social defeat, the fact we knocked down the level attenuated or rescued the phenotype of these mice. Okay? I just want to show another example of using the same technology, the same concept. And this is a field a lot of people in neuroscience use what we call enriched environment. There's a lot of literature both in humans and rodents that the fact if you give mice, kids, through development, meaning from weaning till adulthood, enrich environment will include enrich sensory stimuli, games, for example. They have running wheels, meaning, mice will run between 6 to 7 kilometers per day if you just give them a running wheel. It's a lot for such a tiny mice. They will have 12 friends instead of 3 friends they normally, or 4 friends they normally have. And this is a changing environment, meaning they have house set, the setup is changing every week. And we put them there from weaning, from the age of 4 weeks, till the age of 10 weeks. Okay? Till adulthood. And then we go and compare the anxiety level. And people did it before, and these mice are much less anxious. And it's known those mice can cope much better with any stressful challenge. They have increased neurogenesis in the hippocampus, the other people did. But what interests us was why. Okay, why these mice are significantly less anxious than mice the same. They're brother. Okay, we split them after weaning. This condition or this condition. Okay, and we isolated the amygdala, and we did the whole genome array, the whole 30,000 plus minus genes in the array. There's a lot of genes which changed. One of them in the amygdala was, of course, very interesting for us, and is the CRF receptor type 1. Okay, the one which is responsible for activating the stress response. Okay, we know from pharmacological studies, if we activate this receptor, which in this case in the bilateral amygdala, we increase anxiety. Okay, and these mice showed a robust reduction in the endogenous expression of this receptor. So again, how we can prove that this change in this gene, in this very specific brain region, may be responsible, at least in part, for this phenotype. So again, you can do the same concept. You generated viruses which knock down the level. Don't see it much very good here on the screen, but what we can do then, we can inject them specifically into the basolateral amygdala. So if you punch out this area, you can see we knock down the level, very similarly to what we saw in the array. I'm not showing you again the whole data, But then when you go and phenotype these mice, these mice grew up in a standard condition, not enriched. And we tricked, in a way, the brain. We forced this reduction in this specific brain area, and then went and looked at the phenotype. And these mice are indeed much less anxious. Okay? So again, it's the same concept. There's a lot of study going from the behavior to the gene, and again from the gene into the behavior. I want to give you just one last example on the concept why this type of dissection, this genetic dissection, is important. So the gold standard in this field is that CRF, if you inject it into the brain, and this is a good practice to any lab if you want to do a student lab. You take a mice or rat, inject CRF into the ventricles. It's enough. Wait a few seconds, and you can see what anxious mice look like, okay? He just starts scratching, going to the corner, he doesn't know what to do with himself, he's running, stopping. You can see what a panic type of attack, what anxiety or fear means, which is very nice. So this is 30 years of literature showing that this receptor, the CRF, activating the CRF receptor type 1, is anxiogenic, increasing anxiety. So, again, I'm sorry you don't see it here very well, but as I mentioned, CRF is widely expressed in a lot of brain areas, not only in the amygdala, and one of the highest brain areas expressing CRF is the globus pallidus externa. What globus pallidus to anxiety? Nothing that we thought of. But we did this, and we did a lot of locomotor studies. Nothing was changed. Different locomotion, different challenges. But when we did anxiety, these mice showed an opposite phenotype. These mice suggest that the role of the receptor in this area is actually anxiolytic, is reducing anxiety, not increasing anxiety. And how come? And of course, we did all the genetics, and we sent the paper, and the reviewer said, these people are blind about genetic approaches. Use pharmacology. So we did cannulation to the area, double cannulation, bilaterally, and we did pharmacology. And again, we saw anxiolytic phenotype, not increase in anxiety. It was a long battle. We even came up with a model showing that the striatum is making tons of enkephalin. And enkephalin is actually the highest brain area. If you I don't have it here, you can see it in the paper. There's tons of enkephalin which is released here in the globus pallidus externa. Enkephalin is an anxiolytic drug. And we know we showed that it's actually GABAergic neuron. It's inhibition of inhibition, and actually the CRF is affecting the release of enkephalin. So we have like a putative model. But to make a long story short, eventually it came out because the science paper just came out when we were still in review, showing the same concept, that CRFs could be anxiolytic, not anxiogenic, depending on the neurotransmission system is expressed in. Okay, meaning CRF receptor could be expressed by excitatory neuron, for example, in layer 4 of the cortex, could be in dopaminergic neuron in the VTA, or could be in 5-HT neuron in the dorsal and other cell type, and noradrenergic neuron, the lucus ceruleus. So it really depends where this receptor is located in order to be anxiolytic or anxiogenic. So the overall brain effect, if you just activate the whole brain, is going to be increasing anxiety. But this is not the only role. Each role could be totally the opposite, and this probably area is more responsible for switching off the system. Again, it's system number 1, which is supposed to activate, but is actually also responsible for switching off in other brain areas. So this type of dissection is extremely important for, in my view, if we really want to understand a system, a role of a gene and protein in specific brain area, is trying to be specific as possible in our approaches. I will just jump these two because I see that I don't have more time. I want to show you just these last 2 slides. To get much better temporal resolution, many of our studies now move to use optogenetics tool. You're all familiar with optogenetics, I'm assuming. Again, it's too much light, so you don't see it very well. But the idea, the way we do it is we want to target very specific neuronal population. We don't want to target an area. We want to target only the CRF neurons, for example, which, of course, if you take the central amygdala, there's a lot of CRF neurons, but not every neuron in the central amygdala is CRF positive. Similarly with the bad nucleus or the PVN and so on. So what we do here, we generated mice which express Cre recombinase in the CRF neurons. And we take opsins, we take the channel rhodopsin or the halorhodopsin, this is adeno-associated viruses, which are conditional, okay? They are, you see, they are in reverse orientation. There's the double flux. And when we inject these viruses, for example, into the PVN, they can infect any cells in the area, but they're going to be flipped. They're going to be activated only in CRF positive neurons. Okay? You with me? And then I can manipulate only the CRF cell. I can switch off or switch on this population of neuron while the mice is engaged in a specific task. Okay? And we already have very, very nice data. I didn't put it here yet on manipulating these two brain areas during elevated plasmids, for example. Like we can totally switch in two different projects. We can totally switch the anxiety level of the mice. We can actually cause him not to leave the closed arm at all in one way. And we can, in social interaction tests, we can totally change his decision whether to see a familiar mice or a novel mice. So the system is working very well, and I think it's a wonderful tool to, if you use it smartly enough, because there's a lot of literature out there that is not very specific, but it's allow you to ask very specific questions in very, very good temporal resolution and in behaving mice, which for us it's ideal. I'll finish with this slide, and I just mentioned that we have a lot of interest in early life stress, just because of the concept, what I told you before, because early life stress can definitely shape or program your brain in a way that it will be affect or be susceptible or resilient differently to stressor in adulthood. And I mentioned in the beginning of my talk that the placenta, this is the placenta, you see the embryo and the placenta, that the placenta is expressing huge amount of CRF, for example. And it is a CRF binding protein which is responsible. And there is a really nice studies in humans already showing, placenta is the only tissue it's really, really easy to get from human, Everyone is willing to give the placenta after he's getting the baby. So we have a lot of access to this type of tissues and other people also. And there's really nice correlations, very strong correlation actually, between the level of CRF in the placenta in humans and the chance the mother would develop postpartum depression, in one way, affecting the mother. And the other is affecting the pups, the embryos, meaning the birth rate will be much lower. The chances this embryo, when you're going to be adult, will develop metabolic syndrome, heart-related diseases, and psychiatric disorders, again, is significantly higher. Again, the mechanism is not understood, and what we did recently, we manipulated this placenta genetically, which is a very nice trick, very easy, it's what I want to show you. And again, we do not care really about the placenta. We want to see the outcome, because this is the crosstalk between the embryo and the mother. Okay, but we're interested in the outcome in the brain of the adult, how this is shaping the brain of the adult pups. Okay, so what we do here, we just take blastocyte from pregnant from females, and we just remove the membrane that surround the blastocyte. Okay, it's called the zona pellucida, because viruses cannot penetrate. And if you remember the structure of the blastocyte, we have the trophoblast cells, which make the surrounding, and the inner cell mass. The inner cell mass will be developed with the embryo, and the trophoblast will be developed to be the placenta. So we take these blastocytes, we put them in the incubator for 1 hour with a drop of virus. The viruses cannot penetrate, but they will infect only the trophoblast cells. And then we go and implant them back in the uterus of female. And this is the outcome. So you get only transgenic placentas. Okay? And since we cannot really follow them, because we don't see them after birth, what we do, we co-infect them with the gene, with the virus which expresses luciferase. So we can, using then luciferin and this type of imaging, we can really follow the transgenic placenta and then ask. And we have a very nice study in revision now showing the susceptibility to develop post-PTSD if you overexpressing this type of gene. So I will finish here thanking this outstanding group of students, which I think are really blessed with outstanding postdoc and student and generous funding. And thank you for your time. I'm happy to answer any questions. Thank you. שאלות. Question.
So, the comparison between the globus pallidus and the orbital expression combined with your knockout is last for the hierarchy, which is on top. Did you try to overexpress in the knockout only the globus pallidus?
It's a good point. So what we're trying to do, it's a very good question. So you say if one system is a coping system, it should be activated after the first system is activated. And there's a lot of fundamental questions in the field of stress, you know, that people didn't answer. If I ask a very simple question, what would be, if you look at any of these parameters and you say, okay, I'm going to give you a single stress now, and I'm going to come again after half an hour and give you another stress. Is it the same population of neurons? No one will know. So what we did now, we generated, I don't have it here, a reporter line for the two receptors, meaning we have a green mice for the type 1 receptor, red mice for the type 2. Okay? We cross them, so now they are red and green mice, labeling different receptors. And then we did stress and do kinetic. Okay, meaning then we sacrifice after a few minutes, half an hour, and then we start doing CFOS and ZIF, different early mediated genes. And we're trying to see which population is activated when. My prediction is not going to be, of course, black and white. It's going to be so complicated, and the concept is that we already have some partial understanding on the bed nucleus, which I think is one of the most exciting areas. This is the new amygdala. Everyone is much more complicated. And the lateral septum, which are very important in switching off and these responses. And then we're coming now with optogenetic tools and trying to interfere. Meaning, okay, I know it's activated after such and such time, or going up and down. Then we come with optogenetic tools and trying to block this increase or decrease and how it's going to affect the outcome. Okay, I hope it was clear, but this is where the field is going actually.
So what the CRF actually does locally, I mean, if you overexpress in a neuron, you suppose it then secretes it and affects the local neurons around it, or does it project to other brain regions? So how specific is it?
It's a good point. It's a very good point. I didn't mention it in the beginning, but somebody not familiar with peptides. But peptide will be expressed by the cell body, will be stored in vesicles, will be eventually stored in the nerve terminals. Meaning, if this neuron is going to receive some stimuli, going to be activated, the nerve terminal is going to release this peptide. Okay, this is the way those peptides are acting. When we overexpress it, of course, it becomes much more complicated in a way, because if you overexpress it only in endogenously expressing neurons, it's probably going to behave the same. But what happens if you overexpressing it not necessarily in relevant neurons, because you use a general promoter, which this is one of the problems in this system. But again, every model has its limitation, and this is one of the limitations. This is the reason you're trying to get as specific as possible in the neurons you're manipulated, whether it's going to be with overexpressing or knockdown, or whether you do it with optogenetics. This is the reason I showed you the example of the optogenetics, because we target only endogenously expressing neurons. Okay. One more comment: when you knock down, you don't really care if you infect non-relevant neurons, because they don't have your gene anyway expressed, so it's okay to infect them. It's more affecting the concept of overexpression rather than knockdown. Okay? Question?
You mainly talk about one specific activity. What do you know about how that's activity is done?
The activation? You mean electrophysiologically, what the properties of this? The activation, of course, there's a lot of literature about activation. It's really in which brain area. The PVN, which is the final common site, meaning if you perceive the stress from different perception system can go be strong blood, so you can see something, or you can smell a corpse or something. In all these cases, if we do early native gene activation, the PVN, for example, is going to be always activated because it's receiving inputs from all over because you want to release the cortisol. So, and it's even saturated very quickly. So it's not even the intensity of the stressor is not really important. You're going to maximize the response very, very quickly. But we see that in other brain regions, again, it's not done very much in the literature. It's done for the PVN, but not for the other brain area. And we do it now, again, as I mentioned to Mona. Some brain region are going to be activated only in very extreme stress, what we call a traumatic stress. Some of them are going to be activated only in psychological and physiological. And I give you one really, really nice example. Mona, you will love this one. What we do, what is the difference between someone that's actually is getting hurt, physically hurt by a trauma, let's say a blast, a soldier, and his friend standing next to him, and he's observing the trauma. It's totally fine, wasn't hit at all. So we developed this, we use this model, which you put one mice, they see each other, they can smell each other, and one is receiving food shock, okay, which is a traumatic shock, and the other one is observing him receiving the shock. And then we do have a reminder, never mind, it's a long protocol, you come after a few weeks, and you're just putting them and reminding them of the trauma, not receiving any shock. And then you compare them behaviorally, neuroendocrine, meaning the level of the hormone, and the neuronal activation in the brain. And this is striking, meaning the behavioral, they have these differences in the intensity of the activation. The hormone is going to be activated if you observe the stress or if you really getting a painful stimuli, the cortisol is going to go up. Behaviorally, in anxiety, later, the observer will be okay, better than the one, but still different from control. If you look on the brain region, it's very different. Some brain areas, for example, the lateral septum is like switched off, regardless of whether you observe the stress or you get painful. Some other area will have gradual activation. So we just try to do now to build this network. Where exactly, which is and we of course know the neuroanatomy, so we know which brain region is talking to other brain region. Again, of course, you can imagine it's extremely complex. So we're trying to focus now on some brain region and try to map them. But this is one of the most important questions that people ask now about this field. Okay, תודה רבה, חבר'ה.